Welcome to the deep dive. We're diving into some really cool stuff today. Um, RNA sequencing and all the amazing discoveries happening in the field.
It really is incredible how fast things are moving.
Yeah, it's mind-blowing. So one thing that caught my eye was this article on the GenoHub blog all about the challenges of working with, those older tissue samples.
Oh, yeah, the ones preserved in formalin and embedded in paraffin.
Exactly, FFPE tissues. Researchers have had these tucked away for years.
It's like a gold mine of information, though, right? Formalin does a fantastic job preserving tissues so they can be stored for a long time.
Like a time capsule of medical history.
Exactly. Imagine all the possibilities for retrospective studies going back and analyzing samples from decades ago.
That's what I was thinking. Tracking disease progression over time or seeing how treatments have evolved.
Powerful stuff.
There's a catch, though, isn't there?
There's always a catch.
Getting good quality RNA from those older samples, it can be tough. RNA degrades over time, so getting a clear read on the genetic information.
It's a challenge for sure. But the good news is scientists are developing new library preparation kits specifically designed to tackle this problem.
So it's like having a special decoder ring to unlock those genetic secrets hidden in these older samples.
You got it.
So we're not just limited to fresh samples anymore.
Nope, the possibilities are wide open. Researchers can go back and re-examine tissue samples from past studies, potentially uncovering new insights or even challenging previous conclusions.
That's huge. And this leads us to another exciting development, direct RNA sequencing, or DRS.
Now you're talking. That's a game changer.
I'll admit I was a bit confused about why this is such a big deal. Can you break it down for us? What's the advantage of skipping that conversion step from RNA to cDNA?
Well, imagine trying to understand a complex recipe by only looking at a simplified version.
Okay, I'm listening.
You might miss crucial details or subtle nuances. DRS allows us to capture the full complexity of the transcriptome, including modifications to RNA molecules that were previously invisible.
So we're getting the full picture now, not just a Cliff's Notes version.
Precisely. And that level of detail, it's revolutionizing our understanding of RNA's role in various biological processes.
So there was this study highlighted on the RNA-seq blog where researchers used DRS to investigate how T cells might hold the key to fighting liver cancer.
Ah, yes. Fascinating study. T cells, those are the warriors of our immune system.
Right, the ones that can target and destroy invaders.
Exactly. Researchers at the Karolinska Institute, they used RNA sequencing to analyze T cells within the complex tumor environment of the liver.
And what they found was incredible. They found that certain T cells, both in the liver and nearby lymph nodes, have the potential to recognize and attack liver cancer cells.
It's like giving our immune system a boost, a targeted weapon against cancer.
This means we might be able to harness the power of the immune system to fight cancer more effectively. It's like training your body's own defense force to specifically target the tumor.
It's a powerful concept, personalized immunotherapy.
It's giving new hope to patients who might have limited treatment options. And speaking of targeted treatments, there was another RNA-seq blog post about using RNA sequencing to unlock the mysteries of rare genetic conditions.
Those are often tough to diagnose, right? Caused by these subtle genetic variations that are hard to detect.
Exactly.
But thanks to advancements in sequencing technology, we can now pinpoint those variations with incredible precision.
The blog post mentioned a study using PacBio's advanced sequencing solutions, including FibroSec and Kinnex, or MAS-Sec as it used to be called.
Powerful tools. They allow researchers to look at multiple layers of biological information simultaneously.
So we're not just looking at a flat image anymore. It's like having a multidimensional map of a cell.
Exactly. And by combining genetic sequencing with other molecular data, they can gain a much deeper understanding of how these rare conditions develop and potentially identify new therapeutic targets.
It's like having a high-resolution image instead of a blurry one.
Right, you can see all the fine details that were previously hidden.
And that level of detail can make all the difference when you're dealing with a rare and often poorly understood condition. It's about giving hope to families who have been searching for answers for years.
It really is. And now we're seeing the incredible intersection of RNA sequencing and artificial intelligence. AI is revolutionizing many areas of science and medicine, and its impact on RNA research is truly remarkable.
There was a publication in Nature.com about using deep learning to pinpoint the exact locations of RNA translation within cells, RNA translation. That's the process of turning the genetic information encoded in RNA into proteins, right?
Precisely. Proteins are the workhorses of our cells responsible for almost everything that happens in our bodies.
Building tissues and organs, fighting infections, regulating metabolism, the whole shebang.
It's amazing. And now we can zoom in on where this process is happening within a cell.
So by understanding where RNA translation takes place within a cell, we can gain a deeper understanding of how proteins are made and how this process can go wrong in diseases like cancer.
Exactly. Researchers are now using AI to analyze vast amounts of data from RNA sequencing experiments and identify patterns that would be impossible for humans to spot. This is allowing them to pinpoint the exact locations within a cell where proteins are synthesized and gain insights into the molecular mechanisms of disease. We're basically getting a microscopic GPS guiding us to the exact location of protein production within a cell. It's like seeing where the factories are, how they operate, and what happens when there's a traffic jam or a power outage.
Wow, that's a great analogy. So much going on in those tiny cells. And this level of precision has huge implications for drug development, right?
Absolutely. By understanding how RNA translation is dysregulated in diseases, researchers can develop new drugs that specifically target those errors in protein synthesis, leading to more effective and less toxic treatments.
It's all about precision medicine, tailoring treatments to the individual patient.
It's the future of healthcare. And speaking of targeting specific processes, I came across another fascinating study on bioinf.org that dives into the world of immune responses, specifically in the context of transplantation.
Yes, transplantation's such a life-saving procedure, but it comes with its own set of challenges, right?
Definitely. One of the biggest hurdles is immune rejection. The body's natural defense system can see the transplanted organ as a foreign invader and attack it.
Like a case of mistaken identity.
Exactly. And this study, they were looking at something called allogeneic versus syngeneic splenocyte challenges.
Okay, I have to admit I had to look up those terms.
No worries, it's pretty technical stuff. Basically, splenocytes are a type of immune cell found in the spleen. Allogeneic means from a different individual of the same species, while syngeneic means from a genetically identical individual, like an identical twin.
So they were testing how the immune system reacts to splenocytes from these different sources.
Right. It's like testing the body's security system. Will it recognize a familiar face or sound the alarm when a stranger approaches?
And what did they find?
Well, they found that allogeneic splenocyte challenges triggered a strong immune response, while syngeneic challenges did not. This highlights the complexity of transplant rejection and the importance of finding ways to minimize those immune reactions.
So it's like trying to teach the immune system to tolerate the new organ, to see it as a friend rather than a foe.
That's a great way to put it. And this research could lead to new strategies to improve transplant outcomes and reduce the risk of rejection.
That's really encouraging news for transplant patients. And on a different note, another study on bioan.org caught my eye. This one explored protein binding using a technique called fluorescence measurement. I remember reading about that. Project PRJNA877045, if I'm not mistaken.
You've got a good memory. They were looking at how strongly certain molecules bind to specific proteins, which is crucial for drug development.
Why is that so important?
Well, think of it like this. Proteins are the molecular machines that carry out countless tasks within our cells. And many drugs work by targeting specific proteins, either activating or inhibiting their activity.
So it's like finding the right key to fit a specific lock.
Exactly. If you know how a molecule binds to a protein, you can design drugs that precisely target that interaction, potentially leading to more effective treatments with fewer side effects.
Makes sense. So this study used fluorescence to measure the strength of these interactions, providing valuable insights for drug discovery.
It's amazing how RNA sequencing and related techniques are giving us this incredibly detailed view of the molecular world.
We can zoom in on individual proteins, track their interactions, and even manipulate those interactions for therapeutic purposes.
It's like having a superpower. And speaking of zooming in, there was a fascinating publication in Nature.com about a new method called Instinct that allows researchers to create detailed 3D maps of DNA organization within the nucleus of a cell.
I remember this one. It involves something called spatial chromatin accessibility sequencing data.
Yes.
Sounds pretty complex.
It is a bit technical, but let me break it down for you. Chromatin is the complex of DNA and proteins that make up chromosomes. Accessibility refers to which regions of DNA are open and available for gene expression, basically the process of turning genes on and off.
So it's like looking at a city map and seeing which streets are open and which are closed for construction.
Perfect analogy. Spatial chromatin accessibility sequencing allows researchers to see which parts of the genome are accessible in different cell types and under different conditions. This provides valuable insights into how genes are regulated and how this regulation can go awry in diseases.
and instinct. This is a tool that helps researchers combine data from multiple samples to create a more comprehensive and detailed map of this 3D
You
organization.
got it. It's
Yeah.
like taking multiple aerial photographs of a city from different angles and then stitching them together to create a high-resolution 3D model. This allows researchers to see the intricate details of DNA organization and understand how it influences gene expression.
It's groundbreaking stuff. We're moving beyond just reading the sequence of DNA, and now we can visualize how it's folded, packaged, and organized within the cell. This spatial context is crucial for understanding how genes are regulated and how this regulation can go wrong in diseases.
Absolutely. And staying on the topic of visualizing complex biological structures, another Nature.com publication caught my eye. This one focuses on validating histopathology foundation models using whole slide image retrieval.
Histopathology that's examining tissue samples under a microscope to diagnose diseases,
All
right?
right. And these foundation models, they're essentially AI models trained on vast amounts of microscopic tissue images. They can learn to recognize patterns and features associated with different diseases, potentially aiding pathologists in making more accurate diagnoses.
So it's like having an AI assistant that can analyze those images and provide a second opinion.
You got it. But before we can fully rely on these AI models, we need to make sure they're accurate and reliable. This study focused on validating these models by testing their ability to retrieve similar images from a large database.
So they're giving the AI model a test, showing it a picture, and asking it to find others that look alike.
Exactly. And if the model can successfully retrieve similar images, it indicates a deeper understanding of the underlying pathology. This builds trust and confidence in these AI-based diagnostic tools.
It's fascinating how AI is being integrated into so many areas of medicine.
It's truly transformative.
Mm.
We're moving towards a future where AI can assist doctors in making diagnoses, developing personalized treatment plans, and even predicting disease risks.
It's like having a superpowered medical team.
I'm also intrigued by another Nature.com article that caught my eye. This one uses deep learning and something called a ninja metaheuristic optimization algorithm to predict carbon dioxide emissions.
Okay, the ninja part definitely caught my attention too. I'm familiar with deep learning, but this is a new one for me.
The ninja algorithm is a type of metaheuristic optimization algorithm, which is essentially a sophisticated search strategy used to find the best solution to a complex problem. In this case, researchers are using it to improve the accuracy of deep learning models in predicting carbon dioxide emissions.
So they're using AI to tackle climate change.
Exactly. By combining deep learning with these advanced optimization techniques, researchers are developing more accurate and reliable models to predict and potentially mitigate the impact of climate change.
That's incredible. It shows how AI can be applied to address some of the biggest challenges facing humanity.
Absolutely. From disease to climate change, AI has the potential to make a real difference.
Now, shifting gears back to the world of RNA sequencing, I came across another Nature.com article that delves into the importance of transcriptome size for single-cell RNA-seq normalization and bulk deconvolution.
Yes, single-cell RNA sequencing. It's a powerful tool that allows us to analyze the gene expression of individual cells.
So there are challenges, right?
Of course. One challenge is that the size of the transcriptome, or the total amount of RNA in a cell, can vary significantly between cells.
So it's like comparing the libraries of different houses. Some might have massive collections, while others have just a few shelves.
Great analogy. And normalization is the process of accounting for these differences in transcriptome size so we can accurately compare gene expression between cells.
It's like adjusting the volume on different speakers so you can hear them all clearly, even if some are naturally louder than others.
Precisely. And then there's bulk deconvolution, which is the process of inferring the composition of different cell types within a mixed sample. It's like taking a smoothie made with various fruits and figuring out what percentage is strawberries, blueberries, bananas, and so on.
So by taking transcriptome size into account, researchers can improve the accuracy of both normalization and deconvolution, leading to more reliable and insightful single-cell RNA sequencing data.
Exactly. This research highlights the importance of rigorous data analysis in single-cell RNA sequencing to ensure we're extracting meaningful insights.
It's incredible how we're zooming in on individual cells, analyzing their gene expression, and even reconstructing the composition of complex tissues.
It's like having a molecular microscope that allows us to see the building blocks of life in unprecedented detail.
And speaking of exploring new frontiers, there's another Nature.com article that I wanted to discuss. This one focuses on the spatial integration of multi-omics single-cell data using a tool called CMO.
SIMO, which stands for Spatial Integration of Multi-omics, is a computational framework that allows researchers to combine single-cell RNA sequencing data with spatial information, such as the location of cells within a tissue.
So we're adding another layer of information to those 3D models we talked about earlier. It's like labeling each building to indicate what's happening inside.
You've got it. SIMO allows us to see not only where cells are located, but also what genes they are expressing and how they are interacting with their neighbors. This spatial context is crucial for understanding how tissues develop function and respond to disease.
It's like watching a movie of a city instead of just looking at a still photograph. You can see how people move around, interact with each other, and contribute to the overall dynamics of the city.
That's a great way to put it. SIMO is giving us a dynamic view of biology, allowing us to see how cells behave and interact in their natural environment.
It's amazing how these computational tools are allowing us to make sense of this incredibly complex biological data.
Computational biology is revolutionizing the way we do science. It's helping us to analyze vast data sets, identify patterns, and make predictions that would be impossible with traditional methods.
And speaking of predictions, I came across another Nature.com article that caught my eye. This one focuses on an AI-based prediction of androgen receptor expression and its prognostic significance in prostate cancer.
Androgen receptors, they play a crucial role in prostate cancer development and progression. This research used artificial intelligence to predict androgen receptor expression levels from histopathology images, potentially providing valuable prognostic information.
So they're training AI to analyze tissue images and predict the aggressiveness of prostate cancer.
Precisely. And by identifying patients with high androgen receptor expression, clinicians can tailor treatment strategies and potentially improve outcomes.
This is a great example of how AI is transforming cancer diagnosis and treatment. We're moving towards a more personalized and effective approach to care.
It's an exciting time to be in oncology. We're making significant progress in understanding and treating this complex disease.
And speaking of progress, there was another Nature.com article that I wanted to touch on. This one discusses the selective inhibition of TGS beta-induced epithelial mesenchymal transition to overcome chemotherapy resistance in high-risk lung squamous cell carcinoma.
That's a mouthful. But it highlights a crucial challenge in cancer treatment drug resistance. Epithelial mesenchymal transition, or EMT, is a process where cancer cells become more aggressive and resistant to therapy. TGF-beta is a signaling molecule that can promote EMT. This research focused on developing drugs that specifically block TGF-beta signaling, potentially reversing EMT and making cancer cells more susceptible to chemotherapy.
So it's like cutting the communication lines that allow cancer cells to become more resistant.
Precisely. And this approach holds promise for overcoming drug resistance and improving treatment outcomes in aggressive forms of lung cancer.
It's incredible how scientists are constantly developing new strategies to combat cancer. We're seeing breakthroughs in immunotherapy, targeted therapies, and now even ways to overcome drug resistance.
It's a testament to the ingenuity and dedication of researchers around the world.
Now, shifting gears to another area of medicine, I found a Nature.com article that discusses integrating bulk and single-cell RNA sequencing data to unveil RNA methylation and autophagy-related signatures in chronic obstructive pulmonary disease patients.
RNA methylation is a modification of RNA molecules that can influence gene expression, while autophagy is a cellular process involved in recycling and degradation of cellular components. This research integrated two powerful approaches, bulk and single-cell RNA sequencing, to investigate the role of RNA methylation and autophagy in chronic obstructive pulmonary disease, or COPD.
So they're using RNA sequencing to uncover potential therapeutic targets for COPD.
Exactly. By understanding how RNA methylation and autophagy are dysregulated in COPD, researchers can develop targeted therapies to address these underlying mechanisms.
This is another great example of how RNA sequencing is shedding light on complex diseases and paving the way for new treatments.
It's a versatile and powerful tool that's allowing us to explore the molecular world with unprecedented depth and precision.
And speaking of exploring new frontiers, there was another Nature.com article that caught my eye. This one deals with prompt injection attacks on vision language models in oncology.
Vision language models are AI systems that can analyze both images and text, such as medical images and patient records. They are increasingly being used in oncology to assist with diagnosis, treatment planning, and prognosis. However, as with any AI system, there are potential security risks.
So it's like hacking into an AI doctor and feeding it false information to influence its diagnosis.
In a sense, yes. Prompt injection attacks are a type of cyber attack where malicious code is injected into the input prompts given to these models, potentially manipulating their output.
That's concerning. It highlights the importance of cybersecurity in the age of AI, especially when it comes to sensitive medical data.
Absolutely. We need to be aware of these potential vulnerabilities and develop robust security measures to protect patient data and ensure the integrity of AI-based medical tools.
It's a reminder that as we embrace these powerful new technologies, we also need to be mindful of the risks and take appropriate precautions.
Well said. It's a balance between embracing innovation and ensuring safety.
Shifting gears to another important area of public health, I came across a Nature.com article about HIV/AIDS and HBV co-infection, exploring optimal control strategies and cost-effectiveness analyses using an integer order model.
Co-infection with HIV and hepatitis B virus, or HBV, is a significant public health challenge. This research used mathematical modeling to evaluate different intervention strategies, such as antiviral therapy and vaccination programs, aiming to identify the most cost-effective approaches to control the spread of these viruses.
So they're using mathematical models to simulate different scenarios and determine the best course of action.
Precisely. By considering factors like disease transmission rates, treatment costs, and quality of life, these models can provide valuable insights to guide public health policies and interventions.
This is a great example of how mathematical modeling can be a powerful tool for informing public health decisions and optimizing resource allocation. It's about using data and analytics to make the best possible choices for the health and well-being of populations.
Absolutely. It's about using science to guide policy and make a real difference in people's lives. It's like having a simulator for public health policies. You can test out different approaches and see what works best without any real-world consequences.
It helps us make smarter decisions and allocate resources more effectively. So we've talked about a lot today, from the complexities of RNA sequencing and its impact on various fields to the growing role of AI in biology and medicine.
It's incredible how quickly these fields are evolving and how interconnected they've become.
We're witnessing a scientific revolution right before our eyes. The convergence of genomics, artificial intelligence, and computational biology, it's fueling a wave of innovation and discovery.
It's an incredibly exciting time to be a scientist.
We're not just reading the book of life anymore. We're beginning to understand the language, the grammar, the poetry of it all.
And with each new discovery, we gain a deeper understanding of ourselves, our world, and our place in the universe.
It's a humbling and awe-inspiring thought. So for our listeners who are eager to learn more, what's the key takeaway you would leave them with?
Never stop being curious. Ask questions, explore. Seek out new knowledge. Science is a journey of discovery, and there's always something new to learn.
Beautifully said. And to our listeners, thank you for joining us on this deep dive into the world of RNA sequencing and beyond. We hope you've enjoyed exploring these fascinating topics with us. Until next time, keep those minds curious and keep asking questions. Who knows what groundbreaking discoveries await us in the future?